First, I'd like to thank the meeting organizers for making ISOT 2020 possible. The story I'd like to share with you today is on the wiring of the taste system at the periphery. The fundamental role of the sense of taste is to help us eat essential nutrients, amino acids, carbohydrates, and salts, while avoiding toxic or harmful substances. This really is a matter of life and death, of survival. In this movie, children are eating something for the very first time: anchovy, lemon, yogurt. And we really don't have to be neuroscientists to figure out whether these kids liked what they ate or whether they didn't like it at all. This is because the taste system is hardwired and we show innate and stereotypical behaviors to different taste qualities. In mammals, the detection of tastes begin in the oral tissues before this information is relayed by the ganglion neurons up to the brain. Tastes are detected on the tongue by taste buds. These are the little bumps you see when you look at your tongue in the mirror. Each taste bud contains about 50 taste receptor cells. And after cloning the taste receptors over the past couple of decades, what really emerged is that each taste receptor cell is exquisitely tuned to a single taste quality, such that there are dedicated bitter taste receptor cells, sweet taste receptor cells, and so forth. Taste receptor cells are epithelial cells. After the taste receptor cells detect taste information, this is relayed to the processes coming from the first neuronal station in the ganglia, the geniculate ganglia, which is located behind the ear. Recent work has shown that much like the TRCs, most geniculate ganglion neurons are also specialized to respond to a single taste quality. So we have bitter taste neurons, sweet taste neurons, and salty taste neurons, and so forth. So this forms what is called the label line. Bitter taste receptor cells signal to bitter neurons, sweet to sweet, sour to sour. However, taste receptor cells are epithelial cells. And like many other epithelial cells, taste receptor cells actually have relatively short lifespans. In this experiment, I used the confetti mouse strain to label bitter taste receptor cells using green or red fluorescent proteins and follow them over several weeks. So I would gently pull out the tongue and go back to the same taste bud over and over using a confocal microscope. By doing this, in this taste bud, I was able to see a red cell that appears and then dies off. So this really is a dynamic process. The appearance of a taste receptor cell seems to be a stochastic process. After a bitter taste receptor cell dies, in its place, any of the 5 taste receptor cell types can appear in it, And also what is not shown here is that the nerve fibers from taste neurons are sitting there in the bud, waiting to make new connections with their cognate partners. In any case, the mammalian taste bud is a dynamic structure. Generally, taste receptor cells seem to live for about 2 weeks. So to me, this was a huge evolutionary leap of faith to say that the taste system depends on specialized cells acting as detectors, which are constantly cycling, and yet somehow these cells are at the forefront of a hardwired circuit. How is this done? How do you ensure that the bitter taste receptor cells connect to bitter neurons and sweet to sweet? Imagine what would happen if a bitter taste receptor cell was connected to a sweet neuron. The animal will perceive a bitter substance, which are usually toxins, to be sweet and attractive instead. This would be fatal for the animal. I thought that the taste receptor cells, as they're being born, must express something that will help connections to be formed correctly. So to answer this question, I decided to compare the gene expression profiles from sweet taste receptor cells and bitter taste receptor cells. I chose sweet and bitter taste receptor cells because these are two taste receptor cells that should never cross wire. First step was to engineer mice to label sweet and bitter taste receptor cells with green and blue fluorescent proteins respectively. I dissociated the tongue epithelia from these mice and used a FACS sorter to isolate sweet and bitter taste receptor cells for RNA-seq analysis. We were happy to see that this process worked well because we could see that bitter taste receptors are enriched in the bitter population, and sweet receptor and its co-receptor was enriched in the sweet taste receptor cell population, as we would have expected. So then we next looked for genes that encode known guidance and connectivity molecules. And one of the first things we noticed was that there was a semaphorin, SEMA3A, enriched in the bitter population, and another semaphorin, SEMA7A, enriched in the sweet population. Quantitative RT-PCR confirmed that SEMA3A was greatly enriched in the bitter taste receptor cell population. So what are semaphorins? I'm sure that many of you are familiar with this gene family. Semaphorins are powerful signaling molecules. So for example, in this left panel, there are beads coated with semaphorin 3A, where the asterisk is, and they are able to repel neuronal processes coming from the dorsal root ganglion explant culture. So you can see all the processes are now facing away from the asterisk. There are also cases where SEMA3A can function as attractants. So here, the red cells are expressing SEMA3A, and now you see that some of these processes are going towards the red cells. So we decided to see if SEMA3A has any role in wiring the taste receptor cells. So to determine the role of semaphorins in the taste systems, what we would have liked to do is to directly look at the connectivity of bitter neurons connecting to bitter taste receptor cells and see the effect of semaphorin 3A. However, this was not technically possible. So instead, what we decided to do was to perform in vivo calcium imaging and in geniculate ganglion neurons, and by virtue of that, to infer the connection of these neurons to their partner taste receptor cells. So to do this experiment, we first injected AAV viruses to express the GCAMP6S calcium reporter in geniculate ganglion neurons. Then we opened a small surgical window in anesthetized mice in order to image the geniculate ganglia. We then flowed tasting solutions on the tongue while we were imaging the entire geniculate ganglia. So this way we could use the GCAMP6 fluorescence as a proxy for neuronal activity. Here in this left panel, you're looking at a geniculate ganglion, and as we flow sweet tastings, indicated by the green dot here, you can see that there are neurons that are stimulated by sweet taste. Next, we do this with bitter. You can see that there are neurons that react to bitter presentation. So essentially, we can do this with any of the tastings that we want to test. So we did this with sweet, bitter, salty, sour, and umami, the 5 basic taste qualities. And in a given prep, we typically find dozens of neurons responding to taste and stimuli. And using this, we can infer the taste receptor cell input that a given neuron is getting and to classify them as a sweet neuron, bitter neuron, salty neuron, and so forth. So these are representative traces of 5 different neurons. This neuron was activated by sweet, this neuron by bitter, a salty neuron, umami neuron, and a sour neuron. And so after classifying a few hundred ganglion neurons according to tuning properties, we present the data as a 4-circle Venn diagram. This might look rather complicated, but we really wanted to show the whole picture. Each circle shows the percentage of responders to a certain taste quality from the total number of responding cells. Green shows sweet, blue are the salty responders, sour in yellow, and bitter in red. The outer parts of the circle are the percentage of singly tuned cells, while the intersections show cells tuned to multiple stimuli. The pattern that emerged was that over 90% of geniculate ganglion neurons are singly tuned, responding to one taste quality and only to that taste. So what happens when we remove SEMA3A? When we looked at SEMA3A mutant where SEMA3A had been knocked out in bitter taste receptor cells, we immediately noticed that something was very different. There were so many multi-tuned cells. So if we look at the traces, this first neuron here responded to bitter, sweet, salty, umami, and sour, all 5 taste qualities. The next one responded to sweet, bitter, and sour, and so forth. But when we looked at the Venn diagram, the difference was even more stark. We see a dramatic increase in multi-tuned cells, but importantly, multi-tuning is bitter dependent. So it's within the red circle. So in SEMA3A knockout mice, we are now getting bitter neurons responding to bitters, but also to other tastes. We took this to mean that SEMA3A, which is expressed in bitter TRCs, must be a guiding signal to attract processes from bitter neurons. Once that guiding signal is gone, the bitter processes are lost and they end up making promiscuous connections with other taste receptor cells. To test this, we performed a gain-of-function experiment. We engineered mice where SEMA3A is now ectopically expressed in sweet cells. And if our hypothesis is correct, bitter neurons will now be drawn to sweet taste receptor cells, resulting in an increase of bittersweet multi-tuning. And indeed, that is exactly what we observed. From 2.8% of neurons responding both to sweet and bitter to over 20% responding to sweet and bitter. So in the interest of time, I'm not going to go into detail, but we were also able to show that semaphorine 7A plays a similar role such that SEMA7A is expressed in sweet taste receptor cells and it attracts the sweet neurons to make the correct connections with the partner taste receptor cells. So I think this is a beautiful example of Rogers-Sperry's chemoaffinity code in action in the peripheral taste system. And this opened up many questions, such as, what about the other taste receptor cells? Are semaphorins involved in this? So we looked into this and we found semaphorins that were selectively expressed in salty cells and other semaphorins that were expressed in sour cells, and we're pursuing this. We also tried very hard to completely eliminate bitter-to-bitter connectivity, but it was so hard. And thinking about it, perhaps it makes sense that if bitter detection or toxin detection is so important for the survival of animal, you don't want this to be dependent on one single molecule. So we think that there are redundant molecules in action, and we are looking into this possibility also. But for the next couple of minutes, what I would like to go into detail about is our efforts to go to the next station and identify molecular markers in the geniculate ganglia. We reasoned that if SEMA3A, a ligand, is so important for bitter-to-bitter connectivity, the partner bitter neurons must express SEMA3A receptors. Semaphorins have been studied for a long time, so now we know most of the part of the cognate receptors. So the receptors for SEMA3s are known to be neuropilins and plexin A family of proteins. So we looked at the expression of these cognate receptors, and then we made several mouse lines where to drive Cre recombinases. And we were excited to see this mouse, one of the semaphorin 3A receptors, because the expression was in a subset of geniculate ganglion neurons. And you may have noticed here that this is a Cre line and it's driving GCAM6 expression, because we wanted to see if these neurons in fact responded to taste quality and whether it was selective for bitter. And that really does seem to be the case, because over 80% of these neurons responded to bitters. And with these Cre animals, we're able to do optogenetics. And I'm sure that most of you are familiar with optogenetics by now. This is a method of selectively activating neurons by virtue of light-sensitive channel rhodopsin. So in this experimental setup on the left, I have a thirsty mouse that is waiting for the shutter to open so that he can lick water from his spout. But inside the spout, we placed an optic fiber to shine blue light on the tongue as the mouse is drinking. This is a control mouse where in the absence of Cre, so there's no channel rhodopsin expression. And this mouse will happily drink whether the light is on or off. So this mouse is completely indifferent to the presence of blue light. Now let's see what happens in the semaphorin 3A receptor Cre animal driving expression of channel rhodopsin. So in these putative bitter neurons, we're going to shine light in the terminals on the tongue to see what happens to these animals. And these animals, in fact, are completely disgusted by blue light. When the light is off, the animal is happily drinking. But again, when the light turns on, the animal finds that aversive and will walk away. So I think this is a strong indication that our candidate SEMA3A receptor marker is a good molecular marker for bitter neurons in the geniculate ganglia, and we are excited to pursue this work. And with that, I'd like to thank the members of my lab. I started as an assistant professor at Northwestern University about a year ago, and these are the first few brave people that decided to join a brand new lab. The bulk of the work that I presented today was performed when I was a postdoc in Charles Zucker's lab at Columbia University with our long-term collaborator, Nick Reba at NIH. And most of the work was done with my collaborator, Lindsay McPherson, who's now has her own lab at UT San Antonio. And I'd like to thank Whitehall Foundation for their funding. Thank you.